Hi everyone and welcome back to Advanced Higher Biology, Unit 1, Cells and Proteins. Today we are making a start on Key Area 2, Proteins, and just like Key Area 1, we're going to break them down into the sub-key areas. This is because some of them are very, very large and it'd be a huge amount of information for you to take in just one video. So we're going to start with a very brief one, 2a, the proteome, and we're going to make a start with a term you should hopefully remember from higher biology, which is the genome. So, if you remember, the genome is the total genetic material within a cell. But what we're going to be talking about more often in this area is the proteome. And the proteome is the entire set of proteins that are expressed by that genome. And you can see in this little diagram down here, you can see we have the genome on the left-hand side, all the genetic material in the cell. But there's a much, much larger proteome. There's a much larger set of proteins expressed by that genome. So, there is a reason for this that we touched on a little bit higher, but just to recap, as I said, the proteome is greater than the number of genes, so genome. That's particularly the case in eukaryotes. The reason for that is because more than one protein can be produced from a single gene due to alternative RNA splicing. So you may remember the process of you have a gene, that gene codes for a protein, but depending on what exons are retained and expressed and what introns are removed from that gene through alternative RNA splicing, that depends on the protein being made. So more proteins can be made from just one single gene. That's the reason why the proteome is much greater than the genome itself. The other part that you need to be aware of as well is that not all of those genes are just going to be expressed as proteins. But if a gene is not coding for a protein, then it's going to be doing something else, and we're going to call them non-coding RNA genes. There's two main jobs for these. They can either be transcribed to produce tRNA or rRNA, or they could be molecules that are there to control the expression of other genes. So they have a function, but they just may not all be expressed as proteins in a certain cell type. The final part you need to know as well is that the effect of different factors such as time and different conditions that affect the expression of proteins in a cell type. So there's four main factors that affect the proteins that are expressed. The first is metabolic activity of the cell, the cellular stress, and also a response to signaling molecules. So they all have an effect on the set of proteins being expressed by a cell type. And the final one is fairly obvious. A disease cell is going to have a different set and number of proteins being expressed than a healthy cell as well. So just be aware of those four factors and how they affect protein expression. And that is all for that first very small sub-key area. I'll see you in the next video for 2b, synthesis and transport of proteins. Just make sure you're aware of the genome, the proteome, and the roles of non-coding RNA genes alongside factors that will affect that protein expression. When I see you for the next one, we'll go more into the ultrastructure of a cell, so it might be worth looking over your old notes on cell biology to see what you remember from there. That's all for now. Thanks so much for listening, and I'll see you soon. Bye now.